Welcome to Biodegradable Materials. For my students, this is part of a module on biotechnology and biodegradable materials, but because this is going to be on YouTube, I'm going to separate it into two parts, the biodegradable materials and the biotechnology part. So this is the biodegradable materials. First of all, we have to define what we're talking about. So biodegradable materials, this is number one. Biodegradable materials are things that biodegrade. So obviously there has to be an interaction with biology and it has to turn into something else. It has to be broken down in some way. We can further define it by defining the time that it takes to biodegrade, because if we look at typically what we would consider to be a persistent problem, so something like a chlorinated benzene, which is known to be a problem molecule, that will degrade in the environment over time, eventually. Some of the degradation will be done by biology, not all of it, but some of the degradation will be done by biology, and so we could consider it to be biodegradable. In fact, we will talk about that later in this course. If we consider another molecule, the molecule of lactic acid, polylactic acid, which you might see in a printer. So if you've got a 3D printer, the 3D printer filament, the cheapest type, is polylactic acid. You're supposed to keep it dry, and it is it's sold as biodegradable, but it's actually hydrolyzable. It breaks down with water. So that is a different thing. It's not necessarily biodegradable, although biology helps in that case. So it is biodegradable, but it is also going to degrade on its own. That's also a problem with things like polyethylene, which are not biodegradable in that biology can't attack them in their raw state. But if we give it some energy, some ultraviolet light and some oxygen and time, then it will eventually turn into a state that biology can attack it. So we have to make a decision. The decision in Europe is to define it as an amount of time, which is typically 30 days in a compost heap. And there is a little bit of variation between an industrial type compost heap and a home type compost heap. So if you put it in a home type compost heap, the typical temperature inside a home type compost heap reaches 60 degrees, but it only gets there for a short time. In an industrial compost heap, we get up to relatively high temperatures, slightly higher than that. One can look it up, I can't remember the number offhand. That is important because polylactic acid, for example, has a glass transition temperature of 65 or something, and it softens at that temperature, and it then will degrade way faster above that temperature than it will below it. So it makes a massive difference. If we put polylactic acid into the ground at home, it does degrade, but it takes a really, really long time. The interesting thing about defining 30 days would mean that things like bone and wood, at least hardwoods, would not be considered to be biodegradable. Even cellulose, if we converted it into a block, so we get our piece of paper or our t-shirt, cotton and wood are effectively mostly cellulose. We could dissolve it, recast it into a block and do the biodegradation test, so throw it into a compost heap and wait for 30 days. There's a problem. It would not be gone unless it was a very tiny piece. So we would end up defining quite a lot of things as not biodegradable that we traditionally imagine are biodegradable. So we end up with a span, with a difference in time and a difference in how harsh the conditions need to be. That is useful to us as material scientists because it means we can choose a material that works for the job that we want it for. So if we have an example of a container for a fast food restaurant, then it needs to be able to take relatively high temperatures, and it needs to be reasonably quick to biodegrade as soon as it is exposed to lots of water and air. But it needs to make it home and it needs to survive for a little bit of time, maybe in the fridge. So we have some problems. It needs to degrade reasonably quickly, but we are not too worried about it lasting more than 30 days, because if it's going to be composted, then that's not a massive problem. If we have another example, like stitches in a person, then we would like them to last for a specific amount of time before they dissolve inside the body. And then we have more requirements for what happens to the material, what happens to the parts of the polymer in that case. What do they turn into? Because they have to ideally not cause damage. If we have an implant into, say, a female human being, which contains a hormone to stop them from making eggs so they don't get pregnant, this would be an anti-baby implant. These exist. They go into the hip, and they are polylactic acid, and they will dissolve slowly over time, releasing the drugs. It's excellent because then you don't have to have injections, you don't have to remember to take pills, you don't have to remember very much at all. There are other drawbacks, but theoretically it's a big advantage. There are all kinds of drugs that we might want to keep the dosage relatively constant, because giving it as a high amount for a short time, which an injection typically is either toxic or it doesn't have the effect that we want, because we want a low level of the molecule in the body for a long time. So dissolving it in something and then implanting it and allowing it to degrade over time and release the drug is a good tactic. However, the polylactic acid, as it degrades, gives out lactic acid, which is sour and can cause pain, can cause problems in the local area. So now we've defined what biodegradation is, sort of. Let's have a look at some examples so that we can think about it better. So let's look at a typical bond that we would like to break. It will be an ester bond. So here is our ester bond. This would be in the polylactic acid, for example, where we would have an oxygen here and it would carry on. There's a side chain. So we've got something, let's call this R, and this chain continues in this direction. We don't necessarily need to worry about what's on here. It's this bond that is the ester bond. Those of you in our biomaterials course will obviously be familiar with this bond. Those of you who aren't, maybe not. But we've probably seen it from things: polyester, clothes, fleece, even cotton and biological proteins have a very similar bond. For the rest of people who haven't done so much chemistry, this is a good opportunity to introduce drawing modes, probably, and a few conventions. So we've got our ester here. The R is anything, typically starting with a carbon and then being any group on the end, but it could even be a not carbon. In this case, there has to be a carbon, but very often R just means something attached. It could vary, so we could say underneath R equals a whole load of things. So that is a group. We don't know what it is. On here, for example, let's draw that. On here, for example, there's a methyl group, a CH3 group, and in this style of drawing, I have not drawn the 3D structure, so I haven't drawn the angles of the bonds, and I haven't drawn the hydrogens on at all. So on here, there are 3 hydrogens. So traditionally, my students don't like this style of drawing, but it saves a lot of time, and it is what you will see if you talk to chemists later in your career. You will see chemists not drawing hydrogens unless they are meaningful. So we've got 2 things that I've included, the R and this lack of drawing hydrogens. In this case, we've got our molecule, our ester, and we're going to add water to it and end up with this, which is an alcohol, ROH, is some kind of alcohol, whatever the R is, and we end up with the rest of the molecule, because I haven't drawn it coming to an end here, and this acid group, a double bond O, single bond OH, on a carbon is a carboxylic acid group, so that would be an acid. That would be, if it's making polylactic acid, the alcohol and the acid are on the same chemical, so we would have OH like that, and then on the other end we've got an OH here. So this part is the acid group, and this part is the alcohol group, and the neighbouring molecules connect to each other and form the polymer. I'm going to rub that out though, because what I'm interested in is the mechanism of breaking this, so that we can think about it right now. So in our starting conditions, we don't have a very good reaction. We have water, which is, in chemistry, a very good leaving group, but it's a very bad attacking group because water is happy as water. It's a very stable substance, so it's not very good at attacking things. Then we have our ester group, and this is not particularly easy to attack either. There's a very slight positive charge on here, so we'll put a little delta, meaning a small amount of positive charge on there, and that is balanced by a small amount of negative charge on there. This ether bond, this COC, is not very charged at all. If we take an ester that has two free ends, so it's not a polymer, it's typically not very polar. It's not very much like water. It's more like a hydrocarbon. So those of you who use paint or nail polish will possibly be familiar with the smell of butyl acetate, which is it's an ester. It smells kind of fruity, and it's used as a nail polish remover. It's used as an acetone-free nail polish remover, and it's used in some paints. It behaves like oil, so it doesn't mix with water, and it dissolves things that you can't otherwise dissolve in water, so your nail polish doesn't just come off when you wash. So we have this group, which is reasonably stable and not very good at attacking things, and we have this group, which is also reasonably stable, so that's a bad place to start. We need to do one of two things. We need to either make this group better at attacking, or make this group better at being attacked. Otherwise, the reaction won't go very fast. And so we add, typically, but not always, an acid or a base. The acid one is possibly slightly easier. No, it's not. Let's do the base one first. So the base one is possibly slightly prettier. The acid one is more useful later. So if we put in a base, that means we have some OH minus. Let's put it this way round. So we have some OH minus, which would be, if we draw it in chemistry, we've got a lone pair of electrons which have got a negative charge, and they sit on that oxygen and can do stuff. The stuff that we want them to do is to come in here and attack this oxygen. If they do that, this will cause something to happen, and it's also caused me to have to explain a new thing to you, those of you who aren't from biomaterials, so who should know this anyway. So this is a double-ended arrow, which denotes that a pair of electrons are moving, and they're moving from this oxygen to this point. That means that there will be a charge transfer. So we had a negative charge here, we've got a partial positive charge here and a partial negative charge, which I'll remove because they get in the way and make it too complicated. So now I'm going to transfer the negative charge to here. So let's do that first. It probably isn't a two-step reaction, but let's just draw that. So that is obviously not having organized the shape of the molecule, although it's actually not looking that bad because it should be tetrahedral. So actually, weirdly, it's looking fairly good. This carbon now has too many bonds, and it also has a negative charge, and it resolves that by kicking this bond out, and that is what I'm trying to say. This step happens at the same time as the attack. We don't have time. We can't catch this molecule looking like this. We can catch it looking like this. Now we have a possibility of all kinds of things that can happen, but what's most likely to happen at this point is we'll either take a proton off some water, so we make our OH minus back, or we can go straight into the next step, which is kicking this OR off. So if we kick it off, if this goes in here, we can kick this off, and we end up with our product, which in basic conditions is, so we've got this. Let's draw the rest of the molecule O, so that we've got, so it's clear, this is OH, and this OH comes from, is the OH that we added, and now we've got RO minus, which can now attack some water, because there is water around in base. This can now attack some water, steal the proton, and turn the water into the hydroxy anion, which can now attack our next ester. Because we have regenerated the hydroxy anion, it's behaving like a catalyst, but it does actually take part in the reaction. So in this reaction, the hydroxide does the attacking, and then we get a different hydroxide back. If this OH, or if this O minus attacks a water, that could perfectly well happen, we would end up with a neutral molecule like this, but it's not very stable because this carbon has got way too many bonds on it. So we will eventually, one of these two OHs will attack it back in, and we'll get the same product. But what is important to mention is that there is no reason that this hydrogen should be the original one that was on the OH minus. So those can change. But for practical reasons, it doesn't usually matter. We've usually got water in there, and the water ends up somehow attached. And we end up with this, which becomes ROH, because it steals a hydrogen plus off some water, making an OH minus. So I've reset my reaction, and now I want to come along with some H plus. Remember, we've got a little positive charge down here, a little negative charge down here. So the H plus is going to come along and react with an negative charge that's on this oxygen. The oxygen has two lone pairs, so one of those can attack our H plus. That transfers the positive charge to this oxygen, which can then suck up electrons. So this time round, I'm going to do it all in one. Last time I drew it in two stages. This time we can do it all in one. Let's get rid of those arrows and finish the drawing. So that bond is gone, this bond is gone, and the plus has now been transferred all the way down to here, which is where we want it, because this is where we want to form a new bond. Want being just because logically we know where that's going to happen. So this means that we are doing well with our theoretical reaction mechanism, because we now have the reactive part of the molecule where we expect it to be. Now we can attack this with water. Because we're in acidic conditions, we can't attack it with OH minus, but this species here, this molecule, is now actually positive. It used to have a slightly positive charge down here and a slightly negative charge up here. Now it's got a real positive charge, a whole positive plus, and so that is way more reactive, and it can steal some electrons off this water. Now I need to resolve that. The plus will now be transferred to this oxygen here. So now we end up with this molecule with a plus down here, and it can now regenerate its hydrogen by dropping one of these two hydrogens off here. I'm going to lose this one because it's a bit close to this carbon, so it makes it neater. So if we remove this hydrogen, we can also remove the plus charge because it leaves without its electrons. The electrons stay on the oxygen, the hydrogen loses, so it gets its H plus. We've got our H plus back, and now we have a similar case as we had before. One of these two hydroxides, one of the two alcohols, can attack back in, can collapse, forming a double bond. We don't know which one. while we're at this point, we can say, let's choose this one. Let's choose this one, because it wasn't the one that we started with. Because for the same reason, before the oxygen with the double bond is not necessarily the one that started with the double bond, because we've made it all symmetrical. So this one can now attack in. That's going to make this positive, so we're going to end up with an H plus again. It's also going to make the oxygen have too many bonds, so it will be happy to lose this hydrogen. Happy being not a brilliant operative word. And now we have a choice. We could kick this one off, and then we would be back where we started. So some of the reaction will be doing that. Some of the time we will be going forwards and backwards and just exchanging oxygens or doing nothing. Let's not do that because that's kind of boring. But just to bear in mind that that really happens. So it will be going backwards and forwards doing that. But now we can kick this one off instead. And if we kick this one off instead, we get something new. If we get something new, then obviously we'll be able to notice that something has happened. So if we do that, we end up with, let me resolve this stage. So let's get rid of this H plus here. Let's get rid of this arrow here, draw a bond. Don't know why I closed my pen. Draw a plus down here because we've dumped the charge in. Get rid of this. This has a negative charge on it now. And now all we have to do is transfer this electron to here. And how's the electron going to go? It's going to go into here. So the electron in this bond is going to get eaten by this oxygen. The electron here, which is on the alcohol, but the alcohol anion can jump in here and attack the hydrogen. It doesn't necessarily do that inside the molecule, but if we've got a whole pot of this stuff going on, it can steal it from somewhere else. And if we've got water as a medium, hydrogen ions can be transferred through the water to anywhere. One of the interesting things about acid in water is that it is one of the fastest moving ions possible, and so because it hops between water molecules, and so we then see if we put a hydrogen ion in somewhere into a beaker of water, it is effectively everywhere or instantly. Now you may be wondering, where does the bio come in here? So what we want to do, if we want to catalyze this reaction, is we want to add something up here that can accept electrons with a hydrogen, and we want to add something down here that can attack better than a water molecule. That's how we can make this process work better. So what we need to do is remove electron density from this oxygen by putting something up here that's charged positively. And what we can do here is we can either make our oxygen from our water more reactive by making it into a base, or we can attack it with something else and then replace it afterwards. Those are two relatively common schemes in biology. One of the ways to do this would be to put a metal ion up here. So let's put a metal ion up here. I think it's magnesium usually. Magnesium has a 2+ charge, and it can interact with both of the orbitals on the oxygen, which will obviously make our oxygen really reactive. Magnesium isn't the only ion that we could use for this, and we can look up which enzymes use different metal ions. This is one typical way of doing it. Magnesium, zinc, it depends what size the orbitals are that we want to interact with, and it depends on the conditions and all kinds of other things. We would like our enzyme to fit round our molecule and poke it in the other side with a base, effectively. So we would like some kind of base, and a typical organic base would be nitrogen. So I've looked it up to make sure I get a correct example. I have to admit to my lack of knowledge here, there are a lot of these esterases and they're all slightly different. In this case, we've got a nitrogen base, which is what I wanted, which will take, so this nitrogen is slightly basic, it has a lone pair on it which can attack this hydrogen and so it can be fully happy attached to the hydrogen. Whoops. And that makes this water obviously very much more like an OH minus. We can attach it temporarily like that, and that will make this oxygen have, because we're donating electrons from here, it will make the oxygen have a slightly negative charge, which can attack this carbon, which has a much more positive charge because this electron density in this double bond is all pulled up here by this magnesium ion up here. So depending on which exact esterase we have, we can get a faster or a slower effect, and we can affect different esters. But the general effect is quite similar. We bring something along to this side, and we bring something along to this side, and we do both effectively. Why am I explaining it to you now? It just happens that this is a neat combination of both the acid catalyzed and the base catalyzed mechanisms for ester degradation, and it also happens that polyester and polyamine are the most common types of biodegradable materials. So if we were going to take a protein, silk or something, or just a digestive enzyme, and we were going to eat some food, for example, then we would just change this oxygen for a nitrogen, and all of the rest of the chemistry would be identical, except there's a hydrogen on it. And I can't fit it down there, so I'll have to put it up here. But there's a hydrogen on that nitrogen, and apart from that, the reaction is identical. It behaves very, very similarly, and very many of these esterases and proteases can interact with each other. That brings us to the next topic, which is about specificity. So that will probably be the last thing for today. If we have an enzyme and we are at school, we imagine that the enzyme has a specific target. It does one thing and that thing only. We imagine that an antibody does one thing and that thing only, and we imagine that our body has thousands and thousands of different enzymes that do all of the jobs. We gloss over the fact that when we eat food, we're eating very, very many different bonds of these amides, so of amide groups, maybe some ester groups from sugars, and our digestive system has to deal with that. It doesn't make millions of different types of enzymes to break down specific things. We do have some that are specific, and we have some that are less specific, because in some cases it is better for a molecule to be able to break down all esters. It is better for a molecule that's in our stomach doing meat degradation to be able to break down most amide bonds, because then we only need one. So it is much more efficient to do that, and in reality, in nature, there is a mixture of the two, and neither end is perfect. So on the non-specific end, there are very, very few and not very effective enzymes that will do a whole class of molecules. They're usually better at some than others, and if there are things in the way, then they don't work. You can see here, this molecule has some way of attaching one end to another, and if this molecule in the between happens to have a group that sticks out and blocks this, then it can't grab it. That's typical for polylactic acid. So there are two forms of lactic acid with this group, in fact, pointing in one direction or the other direction. And this arm that connects these two together is on one side of the enzyme, and so it only works for one of them. So if we take a polylactic acid molecule, we can take one form with this arm in one direction, and it will degrade very quickly in a body, and we put it in the other direction, and it takes a very long time indeed because the enzyme can't grab it. So that is specific, but not necessarily helpful. It's useful to us because we can use it for things. If we take the other direction and we want to make it less specific, well, that was making it more specific. So if we want to make it more specific than that, then we would need to make all of the other groups connect better, or we would need to recognize a longer section, but there are always off-target reactions. So in this case, an esterase will break down some proteins. An enzyme that is supposed or normally its normal function in nature, because they're not really supposed to do anything, its normal function in nature is breaking down cellulose, will also break down some other polymers, because it's breaking down this ester bond, or an ester bond with an oxygen in there instead. That's its job in sugar, in cellulose, and so it will also affect other things as well. This is obviously good and bad, but it's important to understand, because it means that if we release something into the environment, there will usually be some reaction of something with it. It may not be very fast, but it will usually occur. So I will leave you with a thought, and the thought is, if we take an ester and we know that its degradation is catalyzed by acid or base, if we put it into our stomach, which is acidic, is that biodegradation? Right, I'll leave that for today. We'll come back some other time.